Welcome everyone. Welcome to our second AfriPopGen webinar of the year. And today I'm excited to introduce Naomi Nicole Biga and Dr. Chiara Battini. Both are based at the University of Leicester. Chiara, floor is yours.
Great, thank you. So yeah, good afternoon, everybody. Thank you very much for coming. And thank you, David, for inviting us to present our work today. We're a big fan of the AfriPopGen, so we're quite happy to be here today. So Naomi and I are going to present you our work in understanding the genetics of smoking behaviors in more than 10,000 individuals of African ancestry. In reality, Naomi, yes. So in fact, Naomi is going to deliver the great majority of this talk, but I'm going to guide you through the beginning of it. So I'm going to tell you a little bit about the story behind and how we met, and then give you a very brief introduction about human genomic variation. And then Naomi will take the lead and present you our project overall. So it's a genome-wide association study for smoking traits in African individuals. She's going to present you the methods and results back to back. So for each result slide, she's going to give you the background of the methods used just before. And then we're going to move, and this will cover the samples included and the phenotypes we look at, the analysis we run in terms of association testing and meta-analysis, the conditional analysis and fine mapping, and then some functional follow-up analysis we have performed so far. And then we're going to move on to discussions and conclusions, and we're going to open up the floor at the end for some questions. So a very brief introduction about who we are. So this is me, I'm Chiara. This is the depiction that Naomi prepared for me. So my background is in biology, and I spent the first part of my education really is in terms of MSc and PhD, doing human population genetic, and I focus mostly on understanding the peopling of Central Africa. And I did my education between Rome and Barcelona. And then in 2010, I moved to Leicester, UK, where I am still now, as a postdoc in genetics. And I've joined the group of Mark Jobling and studied the population genetics of European individuals. And then in 2016, despite staying in the same university, I took a jump of discipline. So I moved from population genetics to genetic epidemiology and joined the group of Martin Tobin, where I've initially been a postdoc and then more recently a research fellow. And I'm now a UKRI Innovation Research Fellow at Health Data Research UK. And my project is about embracing multi-ethnicity in understanding smoking behavior. So it is in the context of this project that I've met all the collaborators that have been involved in the work that we are presenting today. First of all, Michelle and Anya Romuald at the Sidney Brenner Institute, who I've met quite a few years back, and we are involved in a number of collaborative projects together and are here representing the AwiGen cohort. And then more recently, Segun representing the Uganda Genome Resource. But Naomi will give you all the details about the samples involved. So I'm just going to tell you how my path met with Naomi's path. So Naomi has got a background in biotechnology and experimental biology, but she has switched to computational biology during her master's studies, which she undertook at University of Trento. And it is in this context that a year and a half ago, I received an email from this highly motivated student, very enthusiastic about human genetic variation in Africa, who was looking for a project to be performed in the context of an Erasmus program. So I'm very glad I decided to welcome Naomi in my team back then, and she has been with us for about a year now. And she did her Erasmus project in our team, and she's now a research assistant. And has been taking the lead of the analysis of the work we are presenting. So she will present you all the detail of that. So in terms of human genetic variation, we have decided to try and depict some milestones of the incredible development that we have seen in the last 20 years in this discipline. So in 2001, we have the publication of the first draft of the human genome. And then we have picked 3 moments that we think are very relevant to what we're talking about today. And these are the publication of the HapMap project, the 1,000 Genomes project, and the first large-scale whole genome analysis of African populations. And here, these have all contributed to what we are presenting today. In fact, they let us understand how genomic variation is organized in diverse populations in the world, how it is distributed, and how it is within Africa as well. And of course, this didn't happen on its own. There was an incredible development from a technological perspective as well. So we have seen a shift, a development in terms of SNP genotyping technologies, but also sequencing technology. And this was paralleled by an explosion of tools, resources, but also expertise in bioinformatics. So I have 2 figures that I think convey very clearly some of the important messages that genetic has contributed to the liver in the last 20 years. And this is a figure from the latest paper of the 1,000 Genomes project. You're probably all very familiar with these, but what you have here is a pie chart for each of the populations that was involved in the project. And the size of the chart is proportional to the number of genetic variants described in these populations. So you see that there is more genetic diversity in Africa. And then the slices in the pie charts are colored according to where you find the variants. So in gray, you have the proportion of variants that are shared across continents. And colored, you have the variants that are either private to that continent or private to that population. And so what you see here is that the great majority of genetic variation is shared in the world. And there is only a minority of it that is structured from a geographical perspective. So really, this is the take-home message is we're all the same, okay? But we also know that there are some differences among human beings. And despite not being geographically structured very much, but they can inform us in understanding the mechanisms of disease and understanding traits and behaviors, but also our demographic history. So the second figure comes from the GWAS catalog, and it basically summarizes the results of the explosion of genome-wide association studies that we have seen in the last 10 years. So each bar here represents a chromosome, and all the colored dots represent associations that have been described, and they are categorized according to the legend to the right. But beyond the details of what traits have been described, I think the take-home message here, a lot has been discovered in the last 10 years. And genome-wide association studies are becoming increasingly larger with sample sizes that reach up to million individuals. So there is a lot that they have contributed to, and some of these discoveries have informed mechanism of disease, drug development, understanding how people react to specific drugs, and also understanding the risk of disease. But there is an important bias here, and I'm sure you're all aware, but just in case. And this is a figure that comes from a study published in 2019, in which it is reported the population of origin of the individuals included in the studies. And that is what you see on the left of the plot. So it's this histogram here. And what you see on the right is the population of origin of the individuals who live in our planet. And I think it's very easy to see that there is an incredible bias in genome-wide association studies towards individuals of European ancestry. So this is probably not a novelty, but it's important to remember that this is wrong from a number of perspectives. So beyond the historical reasons why it happened, I think it's important to reflect that this does not guarantee a fair representation of the diverse ancestries that we have in this planet. And that means that the discoveries, the incredible number of discoveries that we have seen before, cannot necessarily be transferred across different populations. And that has a high risk of increasing the inequalities that we already see in the world. And another aspect of this bias is reported in these slides. So here we have a pie chart effectively representing the same as before. So these are the individuals included in genome-wide association studies and their population of origin. And again, you see this great bias towards European. And if you see here, the slides representing African individuals, they are about 2.4% of the overall individuals included. But if we look at the associations discovered, including African individuals, we have got a 7% of associations there. So it's almost 3 times as much as the individuals. So there is a disproportionate contribution despite their number being relatively low. And this is even more striking if you look at the individuals of multiple ancestries. And I think the take-home message of this slide is not only is important to include diverse populations in genomic studies for a fairer representation of these populations, but also including the whole diversity that we have in the world will be beneficial to all humans, really, because it increases our discovery power and it will increase our understanding of disease and traits. And so I'm going to now pass you on to Noemi, who is going to explain you the technicalities of genome-wide association studies and the results.
Thank you, Chiara. So here I just would like to start with a theoretical introduction to genome-wide association studies. So in order to be all at the same page, if some of you is not so familiar with this kind of analysis. So genome-wide association studies is a hypothesis-free test for genotype and phenotype association in large-scale dataset. Indeed, the first step of the studies is the collection of phenotypic trait and genetic data. Focusing on the phenotypic trait, this can be a quantitative trait or a behavioral trait. In our case, we deal with 2 behavioral traits, and therefore we divided our individuals in 2 categories, cases and controls. The genetic data instead are usually they usually come from microarray SNPs, and therefore in order to increase the number of genetic variants that we can analyze, we use the imputation analysis that try to retrieve all the other variants present in a genome through the use of imputation reference panel. And so now, since we have both imputed and phenotypic data, we can actually run the single variant association analysis. And then we can visualize the results using a so-called Manhattan plot. So in this kind of plot, we have on the y-axis the minus log 10 p-value of the association. And in the x-axis, we have the chromosome. So and each dot represent the minus log 10 p-value for the association of a single variant. The dashed line represents the genome-wide significant threshold, which is 5 × 10⁻⁸, and it represents the Bonferroni correction for 1 million independent tests. It is usually used also another threshold, which is called suggestive significance threshold, that can be higher or lower than the genome-wide one. And finally, after this primary association analysis, other analyses should be done in order to confirm the results, in order to find biological insights. And among these additional analyses, we can run a meta-analysis that is a meta-analysis performed using individual summary statistics from individual GWAS, and its aims is to increase the sample size and the statistical power. After that, we can retrieve the independent significant variants and then performed affine mapping. Affine mapping is an analysis that tries to find the most probable causal variants in the region described by the independent associated variant. And it grouped these most probable causal variants in a set called credible set. and this kind of analyses are mainly used by Asian approaches. Then we can confirm our results through a replication analysis. In this kind of analysis, we try to replicate the results of our studies in other summary statistics from GWAS run on the same phenotypic trait. And finally, we can find some biological insights of our variants doing different kinds of analyses, such as annotation, gene mapping, or eqtl localization, chromatin interaction. And further, if they are very strong results, further we can go to an in vitro experiment. So after this theoretical introduction to GWAS, I hope that now some of you that were not so familiar can understand better all the analysis. I just want to give some information about the smoking, about the phenotype that we use, so smoking. So smoking is a serious public health threat, and each year more than 8 million deaths are related to smoking. And 80% of the smokers are currently living in low and middle income countries, which mean several countries in Africa. With a deeper look to the African situation, this is a plot that represents the increase of number of deaths in the sub-Saharan Africa, represented by the upper blue line, sorry. And we can see that there is an increase in number of deaths for the period from 1990 to 2016. And also, according to WHO, the death trend on low and middle income country is expected to double by 2030. In this context, the GWAS try to find a genetic component for different smoking behavior traits. One of the most studied smoking behavior traits are smoking initiation, cigarette per day, age of initiation, smoking cessation, and nicotine dependence. Up to now, there are different associated loci confirmed by different studies that involved the dopamine metabolism, the glutamatergic receptors, the nicotine metabolism, and the nicotinic acetylcholine receptors. However, as Chiara explained to you, in GWAS studies, we have a prevalence of European, and also the studies for smoking behaviors are affected by this. So we have a prevalence of European individuals. There are some studies on African Americans, but there are not so many, and also up to now and to our knowledge, there is no study on on smoking behavior traits that include individuals that are currently living in Africa. So the aim of these studies is therefore to perform the first GWAS on 2 smoking behavior traits in more than 10,000 individuals of African ancestry only, who are currently living in 5 countries in Africa and also in the UK. So I would like to start describing a little bit the cohorts that we used, that Chiara already mentioned, some of them. So first, we included the individuals from the African Width In-Depth Partnership for Genomic Studies, or AWIGEN, that I think that most of you knows also better than me. And then we use the Ugandan Genome Research that collect data from the southwestern region of Uganda. And finally, we include the African individuals coming from the UK Biobank. I just want to spend some more words on the AWIGEN cohort, because since these cohorts collect data from 3 distinct geographic areas of Africa, we decided to split this cohort in 3 independent cohorts. And so we obtain AWI East with individuals from Kenya, AWI West with individuals from Ghana and Burkina Faso, and AWI South with individuals from South Africa. So now we define the phenotypes. And the phenotypes are obtained by the combination of answers to specific questions in each cohort survey about smoking behavior. So the two traits that we used are first, smoking initiation. It is a binary trait that compares the non-smokers, the never, versus the smokers, defined as ever. Then we analyze smoking cessation. That is a kind of sub-classification of the smokers, because it is a binary trait that compares who quit smoking, so previous, versus who is a current smokers, the current. So here I just reported a table with the sample size according to each cohort, so the 5 cohorts that we have, and according to gender and phenotypic trait. As you can see from a first look, for female, unfortunately, we do not have we have small sample size, so we decided to go ahead using only males. So now that we have defined the genetic data and the phenotypic data, we can move on to the actual implementation of the association analysis. We decided to use a linear mix model because it allows us to take into account of cryptic genetic relatedness, thanks to the introduction of a genetic relatedness matrix. And we also use different covariates that are age square, and a number of principal components that comes from a principal components analysis specific for each cohort. For each analysis, so we run a GWASIS, so we have 5 cohorts and 2 traits, and we use the implementation of the JAMA software, and we filter for a minor allele frequency higher or equal than 0.001. To evaluate these results, firstly, we measure if our model present any residual genetic bias in a qualitative way through a cuckoo plot, and in a quantitative way through the measurement of the ALDI score regression intercept, which is a measure of the inflection factor that takes into account of the ALDI structure between the variants. And finally, we visualize the results with a Manhattan plot. So for these 10 GWASIS, the results were pretty good. So we decided to use these to perform a meta-analysis. So the meta-analysis actually are 2 meta-analyses. The first meta-analysis was performed on the 3 cohorts that comes from the AWIGEN cohort. So we performed this meta-analysis in order to combine together the results from these 3 studies and represent again the AWIGEN cohort as a whole cohort at the meta-analysis level. So then we use these results to run the second meta-analysis together with the individual results for the Ugandan Genome Research and the UK Biobank Africa Association. For this meta-analysis, we use the modifier random effect as implemented in the Metasoft software. This kind of method allows us to take into account of possible effect size heterogeneity between studies. Also in this case, we did some filters on minor allele frequency, MAC, and info imputation score. And again, we evaluate the results in terms of LD score regression intercept, QQ plot, and Manhattan plot. So now we can have a look to the results, starting from smoking initiation. So from the LD and the QQ plot, we can conclude that there is no genetic residual bias in the model. And from the Manhattan plot, here we have 2 lines. The red lines represent the genome-wide significant threshold, and the blue line represents the suggestive significant threshold that we decided to set at 5 × 10⁻⁶. As you can see, we were able to obtain only association below the suggestive threshold. We can extend this interpretation of the results also for the results that we had from the meta-analysis for smoking cessation. Just one more observation. Both the ALDI score and the QQ plot show a slightly deflated situation. This is expected when dealing with small sample size and binary trait, as in our case. So with these results, we then performed a signal selection of the variants, and therefore we decided to classify the variants according to 3 tiers with different significant levels, combining together the p-value from the meta-analysis and the individual studies. And from each tier, we then define the independent significant variants as the sentinel variants, where for sentinel variants, we define a variant with the lowest p-value in a region of plus minus 250 kb. So for tier 1, unluckily, we did not have any results. But we found 7 variants for smoking initiation and 1 variant for smoking cessation in tier 2, and only 1 variant for smoking initiation in tier 3. We therefore decide to go on, including the variants from tier 2. And for these variants, we performed a conditional analysis and a fine mapping analysis. Here is just a table that describes all the results for the sentinel variants. And I just want to put your attention on the last column, because we checked for the direction of effect in each studies, and they show a concordance in the direction of effect. So just some words about the technicality of this analysis. The conditional analysis aims to find additional associated independent signals, doing an association analysis conditioned on the sentinel variants and take into account the ALDI structure between the variants. So if a variant is shown to be significantly associated in this kind of analysis, it is defined as an additional sentinel variant. And to do this analysis, it was implemented the GCTA COJO software. For the fine mapping, we already seen what it is. So it searches for the most probable variant to be causal, and they group these variants in credible set. For our analysis, we decided to perform the fine mapping in a region of plus minus 100 kb from each sentinel variant. And it was implemented the shotgun stochastic search algorithm using the fine map software. And it was assumed 1 single variant for each region. And we retrieved the variant for the credible set at 99% posterior inclusion probability of containing the causal variant. So in terms of conditional analysis, it confirmed that the independent significant variant were only the sentinel, so it did not found any additional associated variant. And from the credible set and from the fine mapping, we find the credible set associated to each of the sentinel variant. So here we reported in terms of region of each credible set, the length of the credible set, and also the number of variants for each credible set. And we have a total of 2,363 variants that we decided to use for the further analysis. So as further analysis, we use VEP, which is the short for Ensemble Variant Effect Predictor. It is a web online tool that allows us to annotate our variant. And here you have, we can see a plot for with the percentage for each annotation category. And what we conclude and what is expected is that most of the variants are annotated as intronic or intergenic for both traits, about 60% of the variants for both traits. Another analysis that we used is the eqtl analysis as implemented in FUMA. FUMA is the short for Functional Mapping and Notation of GWASs. It is a web online tool that does many different analyses. In particular, we use the SMP to gene function that takes as input GWAS or meta-analysis summary statistics and provides functional notation for SMPs. In our case, we use the meta-analysis summary statistics, and we use the eqtl analysis using 5 eqtl databases. So for the credible set, we were able to find eqtl result for 3 of the credible set. The credible set on chromosome 4 of smoking initiation, in which we found 1 variant affecting the PP2R2C gene in the esophagus musculary tissues. Then in the credible set on chromosome 15, always for smoking initiation, we found 3 variants affecting FBN1 gene in the putamen tissue, that is a region in the forebrain. And finally, we found results for the credible set on chromosome 16 for smoking cessation, in which 18 variants affect the expression of the gene CHES1 in 2 tissues, lung and whole blood. As final analysis, we did some replication and lookup in order to confirm the results of our studies. So the replication was performed using the summary statistics from the GWAS and sequencing consortium of alcohol and nicotine use. This is studies that includes, however, only individuals of European ancestry, and we use the summary statistics available for smoking initiation and smoking cessation. And we use a Bonferroni correction for independent SMPs for both traits, whereas independent SMPs, we use the sentinel variants. For lookup analysis, we try to find if among all the literature associated variants for smoking behavior, some of them were in the region described by the credible set. So for doing this analysis, we use a in-house table of associated variants with 11 smoking behavior traits in 14 studies. And as a result, we found that for the replication, we were able to find a strong evidence of replication only in the credible sets on chromosome 15. And for the lookup, we found 6 variants for the same credible set. So the credible set on chromosome 15 that comes from 4 studies and associated with 3 different phenotypes for smoking initiation and quantity. And these variants were mapped in the region of the gene SEMA60. So taking together all of these results, we ended up on what this research is up today. So we were able to find 4 genes of interest, one that comes from the replication and the lookup, which is SEMA60. And these are just some primary findings about the function of these genes that we found so far. So SEMA60 is a semaphorin. It seems to be implicated in different activities in axon development. It was already found associated with GWAS for smoking behavior traits, as well as for other traits, such as other behavioral traits. The other 3 genes are the gene that we found from the eqtl analysis. So one is FBN1. It encodes for the fibrillin 1, and it has a role in the structural function for connective tissues. Then PPP2R2C, it encodes for the phosphatase 2, and it has a negative control of cell growth and division. And finally, CHES1, it encodes for the carboxylesterase 1. It has a role in drug metabolism, such as cocaine and heroin. It is an approved drug target, and it also may play a role in the blood-brain barrier. So this is our very primary findings and primary findings that we have on this gene. Future analyses are needed. And for example, we need to find if these genes are involved in the same pathways of the already associated genes for smoking behavior. In fact, unfortunately, we were not able to replicate any association already found in the smoking literature. And in fact, another key point of the discussion is the replication strategy that we decided to use. So first of all, I think that we need to say that we decided to use European GWAS for the replication. So in this sense, maybe some of the association are not replicated because we use a different ancestry. And indeed, we are awaiting to be able to replicate our results in the African-American individuals of the Million Veteran Program, because there is a studies for smoking initiation and smoking cessation that include these individuals. And also, we checked for the allele frequency using the allele frequency of the 1000 Genome Project for individuals of the African population and European population for our sentinel variants. And we found that 3 out of the 8 sentinel variants are monomorphic for Europeans. And besides the ancestral differences, we also need to say that with this kind of traits that are behavioral traits, there could be also cultural differences that can influence how the phenotype are defined, and finally can influence the results. So combining all together what we say so far, I just want to conclude with 5 key points. So this is the first genome-wide on smoking traits with individuals living in African countries, in our knowledge. And we were able to discover 7 associated loci for smoking initiation and 1 locus for smoking cessation. However, up to now, we replicated only 1 locus, that is the locus of SEMA60 gene. We found other 3 genes identified through eqtl analysis, and therefore the following analysis will be focused on finding biological implication of these genes in smoking behavior traits. And finally, we are aware that we need to replicate our results in African and African American cohorts as soon as they are available. So I would like to conclude, thanks and acknowledging all the collaborators of these works, starting from the collaborators from the University of Witwatersrand, with whom this project is born even before my arrival, and our collaborators from the Ugandan Genome Research, and all our collaborators and our team at the University of Leicester. And I want to thank you, the founders of the studies, Data Research UK and the Wellcome Trust. And I want to thank you, all of you, for listening and for your time. And we are happy for any kind of comments, discussion, and observation. Thank you very much. I will just stop to share my screen now, so I can see you all.
Thank you very much, Naomi. Thank you for this amazing presentation. It's such an extensive piece of work that your group is able to do in Africa, and such a productive collaboration. Thank you once again for this marvelous presentation. So we're going to open the floor for questions If you have questions, to everyone, if you have questions, you can raise your hand, or you can post your questions in the chat box, and we're going to read the questions aloud for Naomi and Kiara to respond. So I'm just going to start with this question. So looking at where you're presenting from and the population you're presenting on, I would like just ask, why Africa? What's your motivation? Why did you pick African populations? Or is it just that you're just sort of like excited to work on the first GWAS on smoking traits in Africa? So yeah, what's your motivation?
Okay, may I start, Kara? Okay, so yeah, so I think, like, first of all, I think that the fact that we do not have any results on people that are currently living in Africa for smoking behaviors is what is driving this study. So try to find fundings also for this population, also because there is an increase in number of deaths in African countries, and also because there is also an increase in smokers. So it is good to find results, and also after that able to share the results with the public, so to make people aware about the genetic implication of smoking. And yeah, so, and I think, like, since there is this huge majority of studies on Europeans, I think that it's needed to have a fair representation of all ancestry. So Kara, if you want to add your...
I mean, I'm just going to add that maybe there is a component of, you know, an historical component to it. So I do have a passion about African genetic diversity. I think it's an incredibly interesting continent in terms of diversity, both cultural and genetics, really. And the history is so deep that has always been my passion when I was more to like molecular anthropology. And then when I moved into this field, so first of all, African individuals are incredibly underrepresented, and when they are represented, it's mostly in the African American cohorts, really. So the great contribution comes from the States, and that, you know, I think is very obvious that the cultural environment and the legal environment is not comparable between the two places in the world. So that's one aspect, and then the other aspect was an interest in try and you know, look at, so it all started from UK Biobank originally, and then from there it took over to other cohorts. So I was very pleased to see that despite UK Biobank not representing the ethnic diversity that we have in the UK, it did have some individuals that could be grouped into different ancestral groups. And so that's a bit how it started. So partially is a genuine interest in increasing the representation of Africans. Partially is an historical interest and passion on my end. And then, as Noemi mentioned, the World Health Organization is predicting an increase in smoking prevalence in the continent in the next 5 to 10 years. So it's also a matter of public health that will be relevant soon.
Thank you so much, Nigeria, for your contribution in increasing the representation of African populations in GOSs and also in tackling this public health crisis. We have a question in the chat box from this file. So did you consider secondhand smoke? And also, are your study populations all healthy, or did you consider stress or other factors that may contribute to smoking?
Shall I go?
Yeah,
Yes,
it's fine.
So, no, we did not consider secondhand smoke, but it's unlikely that you have a genetic predisposition to that. even though there are some potentially some caveats around that. So you may live in a family of smokers and not being a smoker, so it might still to a certain extent be irrelevant. But as we are looking at your germline mutations rather than somatic mutations, then in that sense is a little bit less relevant. Your second question, so the population, they are not all healthy. I think they do have, I mean, each cohort is a little bit different and has been built in a bit of a different way. So I wouldn't be able to report you the whole distribution of different diseases, but and we have not considered stress or other factors because they were very hard to compare. So the main challenge we had was making sure that we were capturing the same phenotype across cohorts. And anything else you add becomes very hard to compare across different cohorts because the questionnaires are not standardized. So each cohort asks the same question in a slightly different way. And you want to make sure that the phenotype you build out of those questions can capture the same behavior. So that's why we have not complicated the picture further. There are other aspects of phenotypes that we have not touched on. And so Noemi showed you that we've only focused on males. It's not because we don't believe that there are females that have nicotine dependence and use tobacco in other ways, but the use of tobacco is very cultural dependent, and so it was a lot harder to compare again. So that's why we have tried to keep it as simple and comparable as possible. But we are aware of you know, the limitations behind.
Naomi, do you have something to add?
I was going to say that people could also unmute, and I think Alan Christopher was raised hands the other time. I don't know if he has a question.
Okay, yes. Please, Prof. Alan, could you use your question?
No, you're right. No.
Okay. So just as a follow-up to the previous question and Kiara's response, I was going to ask. You said you captured genetic relatedness by computing some sort of genetic relatedness metrics, and what about population structure, broad population structure? I think the other factors that might contribute to smoking and other factors that probably were not accounted for, would they not constitute population structure? So how did you capture that?
Well, we use the genetic relatedness, and we use sorry, we also use PCA analysis as covariates. So this is how we try to deal with a more like general structure for the individuals. Like we try to, so our concept is to use the genetic relatedness metrics for a more pairwise measure for the genetic, and for a more like general structure that try to capture the geographical structure, we use the principal components in the PCA.
Okay, thank you so much. That's
if this was what you were asking, or was it different? And I
think that's it. I think I missed that point, the PCA. And I have raised a question in the chat box. Did you see population structure by study region? This is another question in the chat box.
Yeah, I see. Yeah, well, we were able to find, for example, in the when we, for example, in Acknowledgement, we decided to combine together the. So I'm answering to the other question. Did you see population structure by study region? So, for example, in Acknowledgement, we decided to divide the data right in the 3 geographical area. And for example, are we West? combined data from Burkina Faso and Ghana. And from the PCA of this, we were able basically to have two kind of cluster that represent the population from Burkina Faso and Ghana. So we will still find some population structure, and this is why then we decided to use the principal components as covariates.
Thank you.
Chiara, do you want to add something?
I'm just going to add the comment. So because I believe, so there are a lot of people that are very much involved in the Avigen cohort. Yeah. Thank you very much for being here. So if you have anything to add about Avigen, please step in. But I want to say something about UK Biobank. So we also checked population structure in the UK Biobank individual, which we did mostly to reassure ourselves that we were looking at genuinely individuals of African ancestry. And what we did see is that there is some substructure, but most of the individuals show over like a highly Western African ancestry, really. So the great majority of individuals kind of overlap with the Western African populations of the 1,000 genomes, but not only. So there is a little bit of other groups that you can see. And of course, there are some mixed individuals with Europeans as well.
Thank you so much. Please, does anyone from the Avigen project want to maybe add to the presentation of Naomi and Chiara? And please, you can also raise your hand and unmute yourself to ask a question, as well as you can post your questions on the chat box. While we're waiting for someone to come up, I have this other question. Looking at the presentation, I saw that you didn't really find any significant SNP beyond the genome-wide significant threshold. What probably could explain that, and what are some of the challenges you faced during the study?
Yeah, so we think that, and Chiara, correct me if I'm doing this wrong, but we think that the fact that we had a small sample size that could influence the fact that we found no variants below the genome-wide significant threshold. But however, we then try to use these tier selection that combine together the results from the individuals and the meta-analysis to try to follow up some variants that however, if they even though they do not have the genome-wide significant threshold, they however seems to have a good quality association, and then we with the functional follow-up analysis and also with the replication, we would like to assess even more that even though there is no genome-wide significant association, they still have an important association also in a biological way. So, Chiara, if you want to add.
Yeah, I think that covers it. I mean, some people would tell you that p-value is not everything, and some people would tell you, I'm not going to even look at anything that is not genome-wide significant. I think in this case, we're compromising. And so we have put our threshold a little bit higher in terms of p-value, but we are reassured by the fact that we have combined different criteria and all of the variants show the same direction of effect in all of the individual studies. I mean, that is quite reassuring that they don't seem random associations. So we have decided that that is the threshold we're going to look at.
Thank you. We have another question in the chat box. I missed the array, that's the genotyping you used. Was it tailored to African population?
No, I just want to say you did not miss it. I did not put in the presentation, so sorry if I did not report this information. Yeah, the genotype that were used were specific for the African population, the one for Avigen and the one for Ugandan Genome Research, if I remember correctly, for the genotyping. then the one for UK Biobank is not specific for, thanks Scott. So Scott, right, the Awigen was used the H3A custom array. And for UK Biobank, it was not specific for African population.
I think, may I add something to that as well? So not only, so I think another thing that maybe we should have said more clearly is that we are using imputed data. So this is not only genotype data, so it's not only the SNP chip that matters in this context, but also what the imputation panel we have used.
Yes.
And there is a little bit of a difference in that among the cohorts as well. So we have tried and keep the common variants among all of that. So yeah, so not all of it has been done tailored to the African genetic diversity.
Mm-hmm.
Thank you. Okay.
Thank you for the question.
Okay.
Tesfaye, if I pronounce your name well. And thanks, Ananyo, for the comments about things saying in a right way.
So just one more question before I hand over to David. When you reported on the covariates that you included in your mixed model analysis, you talked about H squared. Please, could you explain what H squared is?
Yeah, okay, sorry. H square is a measure of heterogeneity for the heterogeneity between the studies. And yeah, so it basically tells you if there is a difference between your studies. But in the estimate of the effect size, it does not have an implication. So it is a measure that is reported by the software, and we decided to report it for the sentinel variants, just to report if that variance were measured as a variance with a high heterogeneity between the studies or not.
Thank you very much. David asked, since you used data from 3 main sources, were there any differences in imputation?
Do you want to take this question, Kara?
Well, I mean, the short answer is that we haven't looked at these in detail. I know that Ananyo has been looking at using different imputation panels for Awigen. We have been looked at a similar thing in a different cohort, and I can reassure you that yes, there are differences. Even if you start from the same genotype data and you use different panels, you do see differences. So yes, imputation panel matters. I don't know, Ananyo, if you want to add your results of your comparison between different panels.
Thanks, Kiara. I think it's kind of like, sorry, yeah, it's kind of like, it's different. And I mean, there are a lot of dimensions, but I think we don't have time for this. But yeah, I mean, in short, you said it's very different.
And if I can just ask, this is also why we use the filter on MAF, MAC, and info imputation score in all the data, in all the, sorry, in all the analyses, in order to include good quality data for all the cohort.
Thank you. Thank you so much.
Thank you. You're welcome.
Okay, let's just take this last question, please.
Yes.
Is it possible to differentiate between people who are dying because of smoke from smoking or from simple smoke from cigarette?
Smoke from cooking.
Will I take that?
Oh,
So,
sorry, cooking.
Smoke from cooking and smoke from cigarettes. So I don't think to date there is extensive data in terms of indoor air pollution. Okay, and that is, I have to say, quite nicely asked, because we do have another project that is starting to try and understand what's the best way to actually measure these and what is sustainable on the long run, and how can we conduct a large scale study in terms of air pollution, both indoor and outdoors, in different areas of South Africa, and then ideally expand to other countries in sub-Saharan Africa. So I'm not sure this has been looked at in detail. I don't know if any other person in the call has got this information. As long as I know, air pollution data is pretty poorly captured at the moment in Sub-Saharan Africa, and it's mostly outdoor kind of measurement rather than indoors. So there may be some confusion there, but
okay, thank you so much.